Number 1. The Lacewing larva wearing a backpack of corpses. Picture a droplet of tree resin in a coastal forest 110 million years ago. Inside it, something is moving, or it was, the instant the resin swallowed it. It is barely larger than a grain of rice, and you almost cannot see the animal at all, because it is buried beneath a bristling heap of debris, plant fibers, grains of dirt, and the hollow, drained shells of the creatures it has already killed. This is a lacewing larva, and it is wearing its victims like a cloak. Researchers gave it the name Hallucinochrisa diogenesi when they described it in the year 2012. The team, led by Ricardo Perez de la Fuente, along with Enrique Peñalver, Xavier Del Clos, and Michael Engel, published it in the Journal of the National Academy of Sciences. The specimen came from Spanish amber at a site called El Soplao in Cantabria in northern Spain, from a stage of the Cretaceous called the Albian. Back then, this was a forest of resin-heavy conifers growing near the sea. The behavior preserved here has a name. Entomologists call it debris carrying, or trash packeting. The larva gathers material from its surroundings and piles it onto its own back, holding it in place with long, hooked bristles that you can still see on the fossil. The modern versions of this animal are green lacewing larvae, the ones gardeners call aphid lions or junk bugs. They feed by stabbing prey with a pair of hollow, sickle-shaped jaws and draining the body from the inside, which leaves a perfect, empty husk. That husk goes onto the back. The disguise does two things at once. It hides the larva from the birds and predators that would eat it, and it hides the larva from the ants that guard the aphid colonies it likes to raid. The ants smell their own aphids, not the killer, hiding underneath them. What makes the fossil remarkable is that this is complex, deliberate behavior, frozen in the act. The larva had to reach back and arrange the load. A separate discovery in Burmese amber, reported in 2016, showed that several unrelated insect lineages were doing the same thing around 100 million years ago, which means this is a strategy that evolved more than once. Owlflies did it. Assassin bug nymphs did it. It is one side of an evolutionary arms race, camouflage answering the rise of sharp-eyed hunters in the Cretaceous forest. The El Soplao site itself was only discovered during roadworks in 2008, and it has since become one of the richest amber deposits in all of Europe, yielding thousands of insect inclusions. The lead author, Ricardo Perez de la Fuente, went on to work at the American Museum of Natural History, and later the Oxford University Museum of Natural History, becoming one of the foremost authorities on camouflage preserved in amber. The fern hairs the larva had gathered belonged to plants found in the very same amber, which ties the predator directly to the forest it hunted in. Sit with the image for a moment. A creature smaller than a grain of rice, walking through an ancient forest dressed in the corpses of everything it had eaten. And if a forest could trap something that strange this perfectly, the obvious question is what else it managed to catch. Number 2. Dracula's terrible tick, blood engorged on a dinosaur feather. The next specimen is swollen with blood. Inside a piece of Burmese amber, a tick clings to a single feather, its body bloated to roughly 8 times its normal size, dark and round and full. In the year 2017, a team including Enrique Peñalver, Antonio Arillo, Ricardo Perez de la Fuente, and David Grimaldi published the first direct evidence in the journal Nature Communications that ticks were feeding on feathered dinosaurs. The headline piece shows a hard tick, a species called Cornapalpatum bermanicum, gripping a dinosaur feather. This is the smoking gun, because the feather dates to about 99 million years ago, long before modern birds had taken over. The host was almost certainly a feathered dinosaur, or one of the early birdline animals. In the same study, the researchers named a brand new genus, Dinocraton draculae, which translates roughly to Dracula's terrible tick. And one of those specimens is the engorged one, the one swollen with a recent meal. Now, before anyone thinks of cloning, here is the hard limit. DNA does not survive 100 million years. The blood inside that tick cannot be read like a barcode, and the scientists who found it said so plainly in the paper itself, heading off the inevitable Jurassic Park comparison. What ties the tick to a dinosaur is not its stomach, but its company. The ticks were found alongside tiny hair-like structures, the larval bristles of skin beetles. Those beetles live in the nests of feathered and furred animals today, feeding on shed skin and feathers. Finding them with the ticks suggests these parasites lived in a feathered dinosaur's nest. Ticks turn out to be an ancient lineage, already feeding on land vertebrates by the mid-Cretaceous. The feather the tick grips has a central shaft and barbs, the kind of feather we know from the small meat-eating dinosaurs called coelurosaurs. Modern ticks carry diseases, the bacteria behind Lyme disease and others, which raises a genuinely unsettling question. Were Cretaceous ticks already spreading sickness from one dinosaur to the next? David Grimaldi of the American Museum of Natural History is one of the world's leading amber specialists, and his involvement gave the find serious weight. An engorged tick is special because it proves the animal had very recently been on a living host. There's a shadow over specimens like this, though. This amber comes from Kachin State in Myanmar, a region scarred by conflict, and the ethics of its trade are a real and ongoing concern in science. Hold a picture in your mind. A parasite the size of a sesame seed, fat with the blood of a dinosaur, sealed mid-feast. And if something was drinking their blood, something else was almost certainly living in their feathers. Number 3. The feather-chewing dinosaur lice. Something was indeed living in their feathers. In a piece of Burmese amber, 10 minuscule insects are scattered across 2 dinosaur feathers, and the feathers themselves are damaged, chewed, gnawed at the edges. In the year 2019, a team led by Taiping Gao, Chungkun Shi, and Dong Ren, working out of Capital Normal University in Beijing, described these creatures in Nature Communications and named them Mesothyrus engeli. The name nods to the Mesozoic and to lice, and honors the entomologist Michael Engel. These insects are almost too small to imagine. Their bodies measure between 1400ths and 2300ths of a single millimeter. You would need a microscope just to know they were there. The feathers around them show irregular holes and ragged margins, interpreted as feeding traces, the marks of something eating the feather. Now, these are not true lice in the modern sense. They belong to an extinct group that independently evolved a louse-like way of life, but everything about their body says parasite. They are wingless. Their eyes are reduced. They have stout, chewing mandibles and strong legs for clinging. This is the oldest direct evidence we have of insects feeding on feathers, and it pushes that behavior back tens of millions of years. The feather-feeding niche, the job done today by the chewing lice that infest birds, was already filled by 99 million years ago. That is the heart of it. As feathers evolved, parasites evolved right alongside them to exploit them, a partnership of host and pest stretching back to the age of dinosaurs. The damage on these ancient feathers looks almost identical to the damage modern lice like Menacanthus leave on birds today. Because the tiny nymphs were preserved in life position, actually on the feather, the researchers could argue for feeding behavior rather than mere coincidence. The host would have been a coelurosaur, or an early bird, alive during the great diversification of feathers. The study made its case carefully, using the combination of 2 things: the insects sitting directly on the feathers and the matching damage to those feathers, to argue for genuine feeding rather than mere chance co-occurrence. Dongren's laboratory in Beijing is one of the world's leading centers for the study of Mesozoic insects, and the find slotted into a growing body of evidence that feather-related parasitism arose alongside or very soon after feathers themselves. There is a catch, an open question that still hangs over the find. Every one of the 10 specimens is a nymph, a juvenile. The adult form of Mesothyrus has never been seen, so the full life cycle remains a mystery. Put this next to the engorged tick and the picture sharpens. Dinosaurs were not the clean, glistening models we see in museums. They were living animals, and living animals itch. They were crawling with blood feeders and feather eaters tormented by parasites exactly as their bird descendants are. And the same forest that caught those parasites also managed to catch a predator in the middle of a kill. Number 4. The spider frozen, the instant it struck. Most fossils preserve a body. This one preserves a moment. Inside a piece of Burmese amber, a spider is lunging across its own web toward a wasp tangled in the silk, and the attack is frozen at the instant of the strike. George Poinard Jr. of Oregon State University described it in the year 2012 together with the collector Ron Buckley in the journal Historical Biology. It is the only fossil ever found that captures a spider attacking prey in its web. The amber dates to somewhere between 97 and 110 million years ago. The victim is a male wasp, an extinct species called Cascocilio incassis, and there is a grim irony to it because that wasp was itself a parasite, the kind that lays its eggs inside the eggs of other insects. The predator was about to eat a predator. Around the trapped wasp, at least 15 unbroken strands of spider silk are preserved, which is astonishing on its own, because spider silk almost never fossilizes. Here the web itself is recorded in stone. And there is a second spider in the same piece of amber, which has led to a striking suggestion that these were social spiders living together, a rare behavior to catch in the act. George Poinar is worth pausing on, because his real research into amber and the question of what might survive inside it helped inspire Michael Crichton to write Jurassic Park. The spider doing the attacking is a juvenile. The wasp was alive and struggling when the resin flowed over them both, which is what makes this a true predator and prey encounter rather than two bodies that happened to drift together. From the preserved threads, researchers can reconstruct how Cretaceous orb weavers spaced and anchored their silk, and the detail captured is exquisite, down to the segments of the spider's legs and the antennae of the wasp. It proves that web-based hunting was already a fully developed strategy in the middle of the age of dinosaurs. Not everyone agrees on the second spider. Some argue it was a colony member, evidence of social living. Others think it was an opportunistic intruder that wandered into a web that was not its own. The specimen came from Buckley's private collection, a reminder of how much of this science depends on material held by collectors rather than dug up by universities. The trapped wasp belonged to a family of egg parasitoids, the kind that lay their eggs inside the eggs of other insects, so the web had snagged a predator that fed on the unborn, and that predator, in turn, had become prey. Think about the timescale here. The strike of a spider lasts a fraction of a second. This one has lasted 100 million years and is still not finished. Amber, it turns out, records not just what animals looked like, but what they did. And the next thing it did was catch an animal that had barely begun to live. Number 5. The newborn snake trapped mid-slither. Inside a golden drop of resin lies a delicate string of bone, a spine curled into an S, a baby snake caught in the middle of moving. In the year 2018, a team including Lita Xing, Michael Caldwell, and Randall Needham described it in the journal Science Advances and named it Schaufeus maiamorensis. It is the first and only baby snake ever found in amber. The piece comes from the Hukong Valley in Kachin State, Myanmar, and dates to about 98.8 million years ago. The preserved skeleton is mostly the backbone and ribs, an articulated run of roughly 97 vertebrae, with a total preserved body length of only about 47.5 millimeters. That is shorter than your little finger. The name combines Xiao, for the researcher, Xing Lida, with ophis, the Greek word for snake. And there is a companion specimen, a second piece of amber from the same place that preserves a patch of shed snakeskin, this time from a larger animal. The little snake was either a newborn or a late-stage embryo, an individual that never reached adulthood. Its vertebrae carry a mix of features, some primitive, some more advanced, which has made it valuable in the long debate over how snakes evolved. Just as important is where it lived. The other things trapped in the amber, the bits of plant matter and insects, show this was a forest floor world. Early snakes were not confined to water or open ground. They were living in tropical forests by 99 million years ago. The researchers studied the skeleton using high-resolution scanning, a technique that builds a three-dimensional model from X-rays without ever cutting in to the precious amber. Michael Caldwell of the University of Alberta is one of the leading authorities on where snakes came from. And a juvenile like this is rare and precious because it offers a glimpse of how these animals grew and developed in the Mesozoic. The skull, unfortunately, is poorly preserved, so the identification leans heavily on that distinctive backbone. Researchers still argue over the details. Was this a fully terrestrial forest snake, as the mainstream reading holds? Does it fit better with ideas about an aquatic origin for snakes? Or is it best understood as a transitional form, caught partway through a great evolutionary shift? This discovery also extended the known fossil record of snakes into Southeast Asia and into amber for the very first time. The shed skin in the second piece suggests more than one snake was living in that resin-soaked forest, and it shows these animals were going through the ordinary business of growth, shedding, and replacing their skin in a Cretaceous forest 100 million years ago. Picture it once more. A snake shorter than a finger, frozen before it ever finished its first year of life. And if a whole baby reptile could be preserved this completely, the door was open for something even more delicate, a whole baby bird. Number 6. The hatchling bird, entombed whole. This is perhaps the most complete and the most heartbreaking thing ever found in amber. A hatchling bird only days old, preserved nearly whole, its skull, its clawed feet, its skin, and tracts of its feathers all sealed in a single piece of resin. In the year 2017, a team including Lida Xing, Ryan McKellar, and Luis Chiappe described it, and the specimen earned the nickname Balone. The amber comes from the Hukong Valley in Myanmar and dates to about 99 million years ago. The chick belongs to a group called the Enantiornithines, the dominant birds of the Cretaceous, sometimes called the opposite birds. They ruled the skies alongside the dinosaurs, and they vanished with them. The preserved anatomy is extraordinary. The skull, the neck, a wing, the hind limbs, with their little clawed feet, soft tissue, and feathers, all held in 3 dimensions rather than flattened into a smear on a rock. Based on the development of the skeleton and the stage of the plumage, this bird was only days old when it died. Enantiornithines were what biologists call precocial, meaning they hatched well-developed and ready to move and fend for themselves almost immediately, unlike the helpless, naked chicks of many modern birds. This makes Balone one of the most complete vertebrates ever recovered from amber. Researchers scanned it with high-resolution X-rays to reveal the internal skeleton in fine detail, and the plumage shows a mixture of feather types, including veined feathers and finer, filament-like ones. Ryan McKellar of the Royal Saskatchewan Museum and Luis Chiappe of the Natural History Museum of Los Angeles County are among the leading experts on these ancient birds, and Balone arrived as part of a wave of discoveries in 2016 and 2017 that included isolated amber-preserved wings, nicknamed Angel and Rose. These were not modern birds. They had teeth, and they had little clawed fingers on their wings, archaic features preserved right there in the resin. Because amber captures soft tissue, scientists can even study the microscopic structures that once held pigment, the building blocks of feather color, in a way that bare bones never allow. The same shadow falls here as on the other Burmese specimens, the concern over a commercial trade rooted in a conflict zone, but strip that away for a moment and consider the simple fact of it. A baby bird that lived only a handful of days, its body and feathers intact, suspended and unchanged for 99 million years. A baby bird is astonishing enough, but the next specimen is not a relative of a dinosaur. It is an actual piece of one. Number 7. The feathered dinosaur tail. For the first time in history, a researcher held in their hand not the impression of a dinosaur, not a cast or a footprint, but a genuine 3-dimensional piece of one. In December of the year 2016, Lida Xing, Ryan McKellar, and their colleagues published in the journal Current Biology the first feathered dinosaur tail ever found in amber. The piece was nicknamed Eva. It came from the Hukong Valley in Myanmar, and it dates to about 99 million years ago. Sealed inside is roughly 3.5 centimeters of a young dinosaur's tail, at least 8 vertebrae in a row lined with feathers. The single most important detail is that those vertebrae are not fused together into a stiff rod. The structure birds have, which is called a pygostyle. They are separate, articulated, which proves beyond doubt that this tail belonged to a non-avian dinosaur, not a bird. The feathers show their full structure in 3 dimensions. The central shaft, the barbs branching off it, and the even finer barbules. The coloration appears to be chestnut brown on top and pale underneath, a pattern called counter-shading that helps an animal blend in. The story of how it was found is almost as remarkable as the fossil. The amber had been bought in a Myanmar market in 2015, where traders had taken the contents for a scrap of plant material. Lida Xing recognized what it truly was. The dinosaur was a juvenile coelurosaur, perhaps about the size of a sparrow. Chemical analysis even detected traces of ferrous iron, a chemical ghost left behind by decayed soft tissue and blood. The structure of the feathers offered a clue to one of the great questions in the field, because these feathers were not built for flight. They point instead toward feathers that first evolved for insulation or for display. This was the landmark the moment amber stopped being a source of insects alone and became a source of dinosaurs. Mike Benton, of the University of Bristol, was among those who contributed to the analysis, and the specimen now resides at the Dexu Institute of Paleontology in China. Because the tail is preserved segment by segment, researchers could map the arrangement and color of the feathers along its length, feeding directly into the debate over the order in which feather features evolved. Most likely a small dinosaur became mired in resin, perhaps struggling, and a piece of its tail was left behind. Scientists still debate the deeper meaning, whether feathers came first for warmth, first for show, or first as a step toward flight. One drop of resin held a piece of a dinosaur. Another, impossibly, held the sea. Number 8, the Ammonite and the Seashore sealed in a single drop. Here's a paradox you can hold in your hand. A spiral seashell, a creature of the open ocean, sealed inside a drop of tree resin, a substance that only forms on dry land. In the year 2019, a team led by Wang Bo with Tingting Gu and Edmund Jarzembowski from the Nanjing Institute of Geology and Paleontology published this find in the Journal of the National Academy of Sciences. The amber comes from the Hukong Valley in Myanmar and dates to about 99 million years ago. The shell belongs to an ammonite, an extinct marine cephalopod, a shelled cousin of squid and octopus, the whole group of which died out 66 million years ago alongside the dinosaurs. That is the puzzle at the center of the specimen. How does a sea creature end up in tree sap? The ammonite is a juvenile, identified as belonging to the genus Pusotia, and its presence actually helped confirm the age of the amber. The single piece is small, about 33 millimeters long and weighing around 6 grams, yet it contains roughly 40 individual animals. Some are marine, the ammonite, several sea snails, a clam-like bivalve. Others are creatures of the land and the shore, mites, a spider, a millipede, a cockroach, beetles, flies, and wasps. That mixture is the key. The shell is empty, with no soft body preserved inside, and its opening is packed with sand, which tells us it was already a dead, wave-tumbled shell lying on a beach when the resin engulfed it. Researchers imaged the internal chambers of the shell using X-ray scanning, reading the pattern of its internal walls without breaking the amber open, and that pattern confirmed both the species and the Cretaceous age. So here's the scene the specimen records. Resin flowed down the trunk of a tree growing right at the edge of a sandy tropical beach. As it reached the strand line, it picked up empty shells and grains of sand, and as it continued, it trapped the flying and crawling insects of the forest. This is the first ammonite ever found preserved in amber, and it is one of the most complete snapshots of an ancient coastline ever captured in a single object. It proves the amber forest grew directly against the sea. The grains of sand packed inside the shell are themselves a clue, evidence that the resin made contact with the open beach surface and not merely with overhanging branches. Edmund Jarzembowski of the Natural History Museum in London and the Nanjing team gave us in one golden drop both the ocean and the forest, life and death, predator and prey, and beachcomber, all at once. The specimen lives now at the Nanjing Institute, and it raises the obvious question: how can one ordinary substance trap a beach, a dinosaur, and a hunt with equal ease? The answer begins with what amber actually is. It is not sap, the watery fluid that carries sugars through a tree. It is resin, a thick, sticky defensive secretion that certain trees produce when they are injured or stressed, and it is fossilized resin that becomes amber. The trees responsible were mostly conifers, along with some flowering trees, and they oozed resin to seal wounds and fend off insects and infection. Over millions of years, that resin undergoes a transformation. The light, volatile compounds called terpenes evaporate away, and the remaining molecules link together and harden in a process called polymerization. Fresh resin becomes a halfway substance called copal, and copal, given enough time and the right conditions, becomes true amber. What makes resin such a perfect tomb is its chemistry. It is antimicrobial, and it pulls water out of whatever it touches. It effectively embalms a creature on contact, killing the bacteria that would otherwise rot it and drying the tissues before they can break down. That is why amber can preserve a thing in 3 dimensions, with skin and feathers and color intact, while ordinary fossils flatten a body into a stain on a rock. The animals most often caught are small, soft, and slow enough to be overtaken, or simply unlucky enough to land on a fresh floe. And here is a cruel detail. An animal that struggles often draws even more resin onto itself, sealing its own fate more completely. The same property freezes behavior, which is why we have a spider mid-strike and a larva mid-disguise. Amber does have a bias. It massively oversamples the small forest creatures that lived near resin-producing trees and almost entirely misses large animals, which is why a dinosaur tail is so extraordinary. Modern science reads these specimens with micro-scale X-ray scanning that builds three-dimensional models without ever cutting the amber. Different deposits capture different ages: the Baltic amber of Europe, the Dominican amber of the Caribbean, the Spanish amber of El Soplao, and dominating the Cretaceous record, the amber of Myanmar. Scientists distinguish three stages along the way: fresh and sticky resin, the partly hardened intermediate called copal, and finally true amber, fully polymerized and stable. Trapped alongside the animals, you often find bubbles of ancient air, flecks of debris, and hairs almost too fine to see, all of it sealed in the same instant. A single teaspoon of ancient resin can hold an entire miniature ecosystem, which is why researchers describe amber not as a snapshot, but as a frozen moment, a slice of time held still rather than a still image of it. But that one Myanmar deposit, which gave us the tick and the snake and the bird and the dinosaur, comes with a shadow that science cannot ignore. Nearly every spectacular Cretaceous specimen in this story traces back to a single place: the Hukong Valley in Kachin State in in northern Myanmar. The amber there has been dated with real precision. By measuring the radioactive decay of uranium in zircon crystals from the volcanic rock around the amber, researchers arrived at an age of about 98.8 million years, placing it at the very start of a stage called the Cenomanian, during the height of the dinosaurs. What makes this deposit unique is not just its age, but its richness in vertebrates. Most amber gives us insects. This valley gives us snakes, birds, and dinosaurs. The reason lies in the ancient setting: a lush tropical forest growing right beside a warm sea, dense with life and with resin-heavy trees. Hundreds of thousands of inclusions have now been studied from this one region, and Chinese institutions and researchers, Lida Singh prominent among them, have led much of that work. But the science sits on uncomfortable ground. These specimens almost never come from careful scientific excavations. They are bought from traders in commercial markets, which means many arrive with incomplete information about exactly where and how they were found. Worse, the mining of this amber is tangled up with armed conflict in Kachin State, and the money it generates has been linked to that conflict. In the year 2020, the Society of Vertebrate Paleontology asked its members to pause the publication of new Burmese amber specimens precisely because of these human concerns. It created a genuine tension that has no easy answer between the immense scientific value of these once-in-a-lifetime fossils and the human cost of the trade that supplies them. Museums and journals are still working out how to navigate it. Some specimens may never have the full scientific context that legally excavated amber, like the Spanish and Baltic deposits, can provide. To hold a piece of this amber is to hold 99 million years of distance and a real present-day responsibility along with it. And even when a specimen is perfect, there is a hard ceiling on what it can ever tell us. The single most famous idea about amber is also wrong. The premise of Jurassic Park is that a mosquito trapped in amber, having bitten a dinosaur, holds that dinosaur's blood, and that the blood holds DNA we could use to bring the animal back. The reason that premise feels so plausible is that it grew out of real research. George Poinard Jr., the same scientist behind the attacking spider, did pioneering work on amber, and his studies inspired Michael Crichton. In the 1990s, some researchers even claimed to have recovered ancient DNA from insects in amber. The trouble is that no one could ever replicate those results, and the consensus now is that what they found was almost certainly modern contamination, stray DNA from the lab and the people handling the samples. The chemistry is unforgiving. DNA is a fragile molecule that breaks down steadily after death, with a half-life estimated at around 521 years, meaning half the remaining bonds snap every 5 centuries or so. Even under ideal frozen conditions, the calculations suggest no readable DNA could survive much beyond about 1.5 million years. A 99 million year old specimen is far, far past that limit. There's no usable dinosaur DNA waiting in any drop of amber, and the engorged tick's blood meal cannot be sequenced. What amber preserves is not the genetic code, but the shape, the structure, the surface. It gives us morphology in 3 dimensions: soft tissue, the microscopic pigment structures behind feather color, and, rarest of all, behavior. The ferrous iron found in the dinosaur tail is a chemical trace of decayed tissue, not a strand of code. Sometimes fragments of proteins or pigments endure, and those can still tell us a great deal. The honest scientific position is far less cinematic than the movie, but it is not a disappointment because amber remains revolutionary without a single base pair of dinosaur DNA. The danger lies only in overclaiming, in promising a resurrection that the chemistry forbids. Ancient DNA science has had to learn that lesson the hard way, tightening its standards after the amber claims of the 1990s collapsed under scrutiny. So, if the lesson of amber is not that we can bring the dead back, what is the real lesson hidden in these golden tombs? Lay the specimens side by side and a single living world begins to assemble itself. The blood-engorged tick and the feather-chewing nymphs tell us that feathered dinosaurs were infested, hosting parasites that drank from them and parasites that grazed on their plumage. The lunging spider and the corpse-cloaked lacewing larva tell us about the hunters and the hunted, predators frozen in the act and prey wearing elaborate disguises to survive. The thread running through all of it is coevolution, the endless contest in which every defense calls forth a new attack. The lacewing larva piles husks on its back to defeat sharp-eyed predators. The feather lice evolved in lockstep with the very feathers they consume. Even the trapped wasp was a parasite, a hunter that had become someone else's meal, a whole food web compressed into single drops of resin. The shoreline captured in the Ammonite piece gives us the stage on which all this played out, a tropical forest pressed up against a warm sea. This is what amber does that almost nothing else can. It preserves relationships, not just isolated species, and the relationships it reveals are relentless. The Cretaceous forest was a place of constant miniature violence, the same dramas of hunting and hiding and parasitism that play out in any forest today, simply caught in the act and held still. Parasitism, after all, is one of the oldest and most successful ways of making a living on this planet. And the line from those Cretaceous lice to the lice on a modern sparrow is nearly unbroken. What we are tempted to call horror is mostly just biology seen up close, and seen honestly. Resin caught these interactions entirely by accident, and yet through that accident, we can watch an entire vanished ecosystem behaving exactly as it did 99 million years ago, right up until the moment 66 million years ago, when the asteroid erased nearly all of it. And still, after all of that, the biggest questions remain unanswered. For everything these specimens reveal, they withhold just as much. We have 10 feather-chewing nymphs, but not a single adult, so the full life cycle of Mesophtheris is still a blank. We know ticks fed on feathered dinosaurs, but we cannot say for certain which dinosaurs. The debates rumble on, over whether those were truly social spiders, over where exactly snakes came from, over what feathers were first for. New specimens are now emerging from the Myanmar trade faster than scientists can properly study them, and each new technique opens another door. Higher resolution scanning keeps finding inclusions hidden deep inside pieces that looked empty. New chemical methods keep teasing out pigments and proteins and the subtle pathways of preservation. And the ethical questions keep sharpening, forcing the field to ask how and whether it can study conflict-sourced amber responsibly. Somewhere out there, in unopened lumps of resin, are Cretaceous behaviors no one has ever witnessed, waiting. Each new drop has the power to rewrite a chapter of the story of life, and yet amber will only ever show us one narrow slice of the ancient world, the small things that live near a certain kind of tree. Vast stretches of that lost forest left no trace at all. What lingers, in the end, is the strangeness of the individuals, not a species, but a single chick that lived for days, Not snakes in general, but one newborn that never grew. We have even given them names. Bologna, Eva, creatures dead for 99 million years that we somehow address by name. There's a peculiar humility in holding that much time in one hand, in looking at a single trapped instant from a world so far gone we can barely conceive of it. The forest is gone. The warm sea is gone. The dinosaurs are gone. But the drop remains, and inside it, the moment is still happening. These golden tombs are terrifying, in their way. They are also, and this is the truer thing, windows. The clearest windows we will ever have into a world that vanished long before there was anyone to mourn it.